This presentation is downloaded from bankofbiology.blogspot.com. In this video, I'll be discussing about chromosomal theory of inheritance. Chromosomal theory of inheritance was proposed by Sutton and Boveri in 1902, and it states that the pairing and separation of a pair of chromosomes lead to segregation of a pair of factors they carried. That means if chromosomes are separated, then the factors, or now called genes, are also separated. And Sutton united this chromosomal segregation with Mendelian principles and called it the chromosomal theory of inheritance. So important statesmen of this chromosomal theory were: chromosomes are the vehicles of heredity and they are transmitted from parents to offsprings, and hence the genes are transmitted, and genes are the carrier of characters, and genes are located on the chromosomes. And two identical chromosomes with similar genes form a homologous pair. And homologous pair segregate during the gamete formation, because gamete formation is the resultant of meiosis. And during meiosis, the inner phase, the pair of chromosomes, they separate out. And independent pairs segregate independently of each other. That means that they are not always linked together, means together they are separated, but they can independently segregate. Chromosomals are mutable. Mutation we will study later on. And genes are present on chromosomes, hence they show similar behavior. Genes as well as the chromosomes, they show the similar behavior. When we see the segregation of homologous chromosomes, it is not that the segregation is the same as that of the chromosomes. They can segregate in different, means they can segregate independently. And this, we studied in the law of independent assortment. Here you see chromosome, two colors, red and green, they are together. But if you see here, the red and red are together and yellow and green are together. Here yellow and red were together. So they are having the independent segregation from each other. Now we'll see Morgan's experiment, who worked on Drosophila and proved the chromosomal theory of inheritance. Morgan selected Drosophila in order to do his experiment, and the reason for selection for Drosophila were that they breed very quickly, and life cycle again is very short, that is 12 to 14 days. That means within 12 to 14 days we are getting the results. Throughout the year the breeding can be done, and per mating we get hundreds of progeny, so data is very large, so sample size become very large, and they can grow on simple synthetic medium. And here you can see male and female flies we can distinguish from each other. And Morgan, he did the experiment and he explained linkage and recombination. These are the two terms which we have to know before going to the Morgan's experiment. So linkage means together. When the physical association of two or more genes on a chromosomes, they are placed together, or the distance is very less between the two genes, and hence whenever there is a crossing over, they do not show independent assortment, but rather they go together in the next generation. Hence this is very important statement, that as the linked genes, they linked genes are there, they cannot segregate, they cannot show independent assortment. Therefore, generation of parental combinations are more in the progenies. So linkage, it results in the parental gene combination, and here another term is recombination. The recombination, in this the genes are placed far apart from each other so that they can independently assort, and they form non-parental gene combinations. So linked gene causes parental recombination in the progenies, whereas the recombination produces non-parental type in the progenies. And Morgan carried out several dihybrid crosses in Drosophila to study sex-linked genes. Here you can see in cross 1, he is taking yellow-bodied, white-eyed females. He wanted that the experiment, that the characters are sex-linked. So yellow-bodied and white-eyed females he took, and that was the recessive character. He grew in the laboratory, and another was brown-bodied and red-eyed. They were the males or wild type, and they showed dominant characters. Plus sign indicates dominance over, that means brown body is dominant over yellow and red eye is dominant over white-eyed. And second cross, he took white-eyed miniature winged female and red-eyed large wing or wild type as male. And similarly, the wild characteristics, they were dominant over the created males or females Drosophila in the laboratory. Here you see cross A and cross B. In cross A, parental type yellow and white. Here you see the links are very closely placed together, whereas in cross B, white and miniature wings, you can see that the links are quite apart from each other, and there is no gene on the Y chromosome. So males show the characteristics whatever the genes are present on the X chromosome. Here these are linked genes. So if you see the F2 generation, you will get that parental type are 98.7, whereas recombinant types are only 1.3%. That means it is quite deviation from the Mendel's law, and parental type are quite high. Whereas in cross B, the parental type is 62.8% and recombinant type is 37.2%. And you see here the links are far apart. That is why we are getting the recombinant or the non-parental type much more as compared to the cross A. So this was about the recombination and the linkage, and Morgan now intercrossed F1 progeny, and he found that two genes did not segregate independently, and F2 ratio deviated from 9:3:3:1, and genes were located on the X chromosomes. And when two genes were situated on same chromosome, proportion of parental combination was much higher than non-parental type due to linkage. So in the cross, we studied that tightly linked genes show less recombination and loosely linked genes show high recombination. Now, modern projects, they are all based on the genetic maps. When we say study the linkage or recombination, we find that the recombinants are much more when the links are far apart, and this can be used in placing the genes on the chromosomes, and that is known as mapping, gene mapping. Gene mapping is the measure of distance between the genes and their position. Hence, we can locate the genes on the chromosomes. And in the next video, I will be discussing about the sex determination. Thank you.